Hi, you guys. So I'm going to go quickly over some things that we have to go ahead and that we have to do to go over some of these topics that we went over this semester. So this is just a quick review of everything we've learned this semester. So first of all, speciation. So going back to our evolution deal, right? Species is a group of organisms that can create fertile offspring, which means that in order to be a species, you have to be able to go ahead and to survive and reproduce another organism that's fertile. And again, that excludes anything that is not fertile, such as a mule or anything like that. Um, New species can be created over time due to a couple other things. They can go ahead and they can go through reproductive isolation, which means that they're not able to reproduce with each other. And that kind of goes into some of the having different mating behaviors, being separated by space, anything like that is reproductive isolation. You can also be separated by the different times that you mate as well. Natural selection, so going ahead and picking which animals are going to survive. So kind of like the animals going ahead and surviving. So you have the natural selection here, which is the insect eating one. The woodpecker type eating insect and the seed eating ones, over time, different beaks would probably develop into different ones that would be able to survive. Okay. Next thing, if I can get this. You have a little bit more of natural selection, right? So you see this hawk, it goes ahead and it's able to see these lighter mice. So since it's able to see those lighter mice, it's going to go ahead and it's going to select, and the population will become more black over time to go ahead and to hide. This whole giraffe thing, the giraffe that is able to reach the leaves are the ones that are going to survive because of the fact that these giraffes right here are able to get the food that is going on up here, right? It can't get the food. It can get this food and it can get that food versus this giraffe right here that's way too short, right? That giraffe you'll see will die later because it runs out of food basically. Then we have relative and absolute dating. So this is the dating of fossils. Relative dating is we're going ahead and we're comparing different rock layers, and we're saying that rock layers are older than another, right? We're not given exact ages. We just go ahead and say, oh, this rock is youngest and this rock is oldest because it's younger because it's on top, right? Numerical dating or absolute dating is when we actually give some sort of number to our dating. So you guys will see that over here we have our radioactive dating, which we're separating things by half-lives and all of that. And we have exact numbers. So after 1.3 billion years or after 2.6 billion years, that is what we see. Versus relative dating, we go ahead and we age fossils based off of what we see from each other. Okay, so just some evidence of evolution rundown sort of deal. So we can have this whole natural selection thing going on. So we have these predators that are going ahead and helping select which animals are going to be able to survive. We have homologous structures, right? So homologous structures means that we are going to go ahead and we have the same sort of deal, but they have different functions, right? And then we also have analogous structures as well. We have all of these molecules, this molecular evidence. So how similar they are in amino acids indicate how closely they can be related. So now that you guys know what this stuff means, you guys will see that the differences in one or two amino acids is not as much. You guys will also see some of the fossil evidence. We have some of the vestigialist structures that you guys will see in the whale, such as the pelvis in the whale and all of that. And then embryos, right? So looking at these things at very, very early stages. If you guys remember doing this, you guys will see that the very, very young ones kind of look very similar to each other. Okay, so next thing, epigenetics. So epigenetics is when it's above the genome, right? And that means when we go ahead and we turn genes on or off, right? So when genes are on, they go ahead and they go through transcription. When a gene is off, it does not go through transcription. So you have all of these little molecules that are on your DNA going ahead and turning genes on or off, right? What turns genes on and off are the following, right? So lifestyle basically turns genes on or off. So if you're exposed to diseases, if you're exposed to certain diets, right, psychological, social interactions, right, medicines, all of that can influence your epigenome and can influence what is happening on there and what genes you will express over time. So that's kind of what we meant by with the twins looking different over time. Okay, so this goes back into Punnett square. So dominant versus recessive. So the main thing to remember with dominant versus recessive is a dominant allele is an allele that is going to show, right? Because the allele is going to be expressed no matter what. And again, we're talking about complete dominance in this case, right? A recessive allele will only appear if there's two recessives there. If there's not two recessives there, then it won't appear. So you guys will see that we got these cats going. When you see a capital letter, that means it's dominant. So I have a dominant, brown is dominant over gray. So you guys will see that this cat is going to be brown because it has two capital letters. You'll see that this cat has one capital, one lowercase, and since it's complete dominance, it will still be brown. This cat is gray because it has two lowercase letters. So those are some of the things that you guys see on there. Punnett squares, very simple. You guys go ahead and you set up your parents all the way out here, right? So on the top, you guys set up your parents. After that, you guys determine if they're homozygous, if they're heterozygous, if there are homozygous recessive, all of that, and you set that up. And then you guys go ahead and you bring these letters down, as you guys saw on here, right? And then you bring these letters across, as you guys see on there. So you guys see that you bring those letters across. And then that is how you go ahead and you solve the Punnett squares. And again, you guys have to solve each percentage, and each box is worth 25%. Okay. Next thing, sources of variation. So why things can be different from each other. The main thing is that we can have a couple of different things happen. The main thing that you guys need to know is that a big source of variation happens in meiosis. In meiosis, we go ahead and we separate and we create sex cells by separating our DNA that we have into half, right? Because we get 50% from mom and 50% from dad. So what will happen is when these chromosomes go ahead and they separate, they trade pieces of DNA with each other. So then you create 4 unique sex cells. So that is pretty much a main source of variation. The next thing is that being exposed to different things over time can also change your DNA, like radiation, chemicals, and infectious agents. Those things can be going ahead, and those things can damage your DNA over time. But that has a little bit more to do with the epigenetics sort of deal. So those are some of the things that you guys will see on there. DNA versus RNA. So DNA is this guy right here. So this DNA is going ahead and it's double-stranded. You have thymine, cytosine, adenine, and guanine, right? Versus RNA, this is a single-stranded guy, right? We have uracil, cytosine, adenine, and guanine. You guys will notice that we have uracil instead of thymine. That's the biggest difference between the two. You guys have to know what things are in which, right? So you guys got to know what nucleic acids are in which type. Okay, next one is protein synthesis. So protein synthesis is the whole central dogma of biology, right? We go from DNA to RNA to protein, right? So how we get there is from DNA to RNA, we get there through transcription. That mRNA is able to sneak out of that DNA jail, and it comes out into the cytoplasm. it's going ahead and it's being read by ribosomes, and then this ribosome creates these amino acids, and then the amino acids fold, and we go ahead and we create our protein. So that is pretty much the main thing that you guys need to know. From here, we're going from